Su atención, por favor. Su atención, por favor. Le informamos que una de las charlas será impartida en idioma inglés. Si usted requiere un equipo de traducción simultánea, puede obtenerlo fuera de este auditorio con una identificación. Repetimos, la charla de esta tarde será impartida en idioma inglés. Si usted requiere un equipo de traducción simultánea, puede obtenerlo afuera de este auditorio con una identificación oficial.
We have plenty of time. Tomorrow at what time is your plane leave?
Early evening, 6.
Okay, good.
We have plenty of time. I can talk until then.
Good. And we can
The
discuss
cleaning
science.
lady will not be happy.
Yeah, well, it can remain like that. It
I
really
cannot
doesn't matter.
satisfy everybody.
As long as you are satisfied yourself,
The
that's
Minister
important.
of culture, of unculture.
Burra.
So you saw her, where did you see her dress?
Oh, it made a scandal worldwide because people in Mexico are very, very keen on having peace developing between all the sectors.
But you know, if you look at the dress very carefully, there is no Jewish thing on it. It's the Arab mosque.
That's true.
The church.
Yeah.
The walls that were built by the Turks.
Yeah.
It just from here in Jerusalem, but
Yeah.
but
No,
anyway, there was not even one Israeli movie in this festival that was on the list of competitors. There were no lists. So they told me, what is she celebrating? I said, this is what she's celebrating, that she could eliminate all these leftist peace lovers.
Yeah. Which is the most important part, the most attractive part of Israel society nowadays. So you will have the translation there. I'll make a brief introduction.
In Spanish?
In Spanish, yeah, because this is the Colegio Nacional.
It's okay.
And
But I won't speak Spanish.
no, it's not, no problem.
My Spanish poquito.
It's just, people want
Once
to
I
see
knew
you.
better. Once I could, I drove.
You took classes or?
I did it in Valdivia, in Chile. I did the course.
Oh, course. In Spanish?
No, I spoke English slowly, but they asked in Spanish and I answered.
No, here.
I couldn't speak, but I could get the sense.
Here I would say that about 80% of the audience is fluent in English.
They must.
Yeah, of course, if you want to be a scientist.
Near America,
Yeah.
even in Tijuana.
Yeah. Okay, so we can start.
Yes, of course.
Buenas tardes, a nombre del Colegio Nacional, en especial a nombre del doctor Francisco Bolívar, del doctor Eusebio Juaristi, del mío propio. Para mí es un honor enorme darle la bienvenida a la doctora Ada Jonat, que es una científica muy distinguida, y a ustedes por asistir a esta última sesión de las jornadas que hemos dedicado a examinar el mundo del RNA. La doctora Jonat viene del Instituto Weizmann en Tel Aviv y es una científica extraordinariamente distinguida que en el año 2009, precisamente en el bicentenario del nacimiento de Darwin, recibió el premio Nobel por su trabajo en la cristalización del ribosoma. Los que ya lo saben por su formación, los que hayan asistido la semana pasada a las charlas que tuvimos en torno al mundo del RNA, estarán perfectamente conscientes de que el ribosoma es una de las estructuras subcelulares más antiguas, más conservadas y más complejas que conocemos. Tanto es así que cuando la doctora Jonat anunció hace ya muchos años su intención de cristalizar el ribosoma para poder conocer la estructura terciaria y ver los sitios funcionales del ribosoma, que es donde, como todos ustedes saben, se lleva a cabo la síntesis de proteínas. Su idea fue recibida con un enorme desdén y hay una serie de anécdotas que no estoy seguro que si ella contara, pero que incluían, por ejemplo, el tener que dormir en el estacionamiento a escondidas en el coche para que cuando nadie se diera cuenta de estar trabajando a deshoras en cristalizando el ribosoma. Bueno, lo logró y ese logro no solamente ha sido absolutamente extraordinario desde un punto de vista teórico, desde un punto de vista funcional, sino también desde un punto de vista evolutivo, porque una de las razones que nos llevó a muchos a acercarnos específicamente a las propuestas de la doctora Jonat fue su insistencia absolutamente correcta de que en el ribosoma estamos viendo un sistema, una estructura que viene de muy tempranamente en la historia de la vida en la Tierra y en la cual podemos ciertamente reconocer algunos rasgos esenciales de la vida muy temprana en el planeta y de su evolución posterior. Dicho eso, de verdad que es un enorme placer personal, es un enorme honor para todos nosotros, los que hemos organizado esta semana, estas jornadas sobre el RNA, y es una distinción ciertamente para el Colegio Nacional, la asistencia de la doctora Jonat, que muy amablemente aceptó venir desde Israel, de donde llegó anoche y a donde regresa mañana. Yo quiero subrayar esto porque es una muestra de amistad enorme y desde luego, y esto no es trivial, a la motivación que le inspiran sobre todo los jóvenes dedicados a la ciencia en este país. Ada, if you please. You can speak from here, you can speak from over there.
Thank you for coming and for listening to this wonderful introduction. I have to fix one of your mistakes. I didn't arrive last night, I arrived this morning at 4 o'clock in the morning. And I would like to tell you that Dr. Lascano and me are really good friends. He discovered me before the Nobel Prize did.
It's true.
That's true. Thank you. So now for the lecture. I wanted to understand protein biosynthesis, how proteins are being made. I will come back to it. It's the introduction. But I learned a lot about RNA, much more than I expected, and this is what I will talk about today. So from the beginning, the central dogma was the genes are in the DNA, and when a gene has to be translated, the DNA itself has the language here, the letters of the language here inside the double helix. It's not available. They cannot be read because of the structure of the double helix that hides them. So first of all, the part that is coding for the gene that has to be translated is being transcribed to messenger RNA, to an RNA molecule. I think you all know what is RNA, I don't have to explain, but here, what is important is that the bases are available. They are on the surface, and they can be translated by the ribosome to the growing protein. And I think you know all of it, that you know that proteins have their sequence dictated by the gene, and the sequence also is dictating their structure so they can do their job. So the sequence is really very important. All ribosomes in all cells, ribosomes exist in every living cell, in bacteria, in cockroaches, in fish, in flowers, in human, whatever, everything that lives. They are universal. It means they did grow, they did evolve with evolution, but the way they do their job, it means peptide bond formation and decoding. This is done exactly the same way, or almost exactly the same way, in all ribosomes. There is a huge number of ribosomes in every living cell, even in bacteria. 80,000 to 100,000 ribosomes work in a single cell. In human, in the liver, maybe 5 or 6 million ribosomes can work together. In vivo, the ribosomes are very, very fast. They act all the time. They can form up to 40 peptide bonds in 1 second. It means a protein that is 250, 300 amino acids is being formed within a few seconds. And they hardly make mistakes. Even if they make mistakes, they get rid of the mistakes before we human beings can find out about them, can detect them. So just for the speed, I was a very good student and the fastest in my class. We had to make 1 peptide bond in second-year chemistry. And I told you I was the fastest. A single peptide bond took me 6 hours. And I can make 40 in a second. So before we go into the lecture, let me show you what we know now about ribosomes. It means what we knew already in 2001, after the first structures of ribosomes were determined, 2 by us, small and large subunits, 1 by Ramakrishnan, the small subunit, and the large subunit by Stites, and there were some lower resolution structures. I accommodated all of them in 1 movie. It means if I saw some motion, I integrated it to one movie so you can see since each structure was determined at a specific functional state, you can see the function. I just want to tell that ribosomes are made of 2 subunits. Each of them is independent in the cell, and they come together when they have to start. And there are cellular factors that control ribosomes, like initiation factors that initiate, and you will see also elongation factors that allows the protein to be built, and termination. So here there is the small subunit, small ribosomal subunit, and messenger reaches it. And look, it takes it. Look now, it takes it. You see? Choop, it took it. It looks very small motion. It's about 7 angstrom, the size of the messenger. And the thing that has to move has a molecular weight of 250,000 Daltons. So it's not so small, it's just small. It looks small, but it's energetically demanding. So here messenger is now in its path, and the whole thing is monitored by initiation factor number 3 that sits here and makes sure everything is fine. Once everything is fine, a tRNA is brought. Maybe I will say now there are 3 sites of tRNAs. tRNAs are the molecules that bring the amino acids. So a tRNA, I'll come back to it later, but here it is, the blue, and it's carried out by another initiation factor 2. So here the tRNA is now happy, sitting in place. The messenger is in place, so the factors can live, and the large subunit can reach. It will associate with the small one based on their surfaces, complementarity of their surfaces, but also to make sure that the association is perfect and very accurate. There are bridges that are being made by the 2 subunits, like here. There are 13 like this by conformational changes. So now the active ribosome has been created. And you will see how it works, the large subunit on top, the small down, and the amino acids are brought by tRNAs with factors that are called elongation factors. So they come in, they are being decoded, make the bond, and go out, and the ribosome helps. In, out, in, out, in, out. These are hands of the ribosome made mainly of RNA. So we took away the large subunit. You can see the decoding being here, and peptide bond being made here, and the newly born protein emerges out of the ribosome through a tunnel. The tunnel is spanning the large subunit. So now the protein is being made here, and it becomes longer and longer. It's now longer than the tunnel, can come out and fold. And the whole thing, the whole process continues until there is a stop codon that says protein is complete. And then factors come, like release factor or recycling factor that replaces the tRNA. You see this gold. The 2 subunits, the messenger, the tRNA, they're all ready for the next protein. The protein itself can go to where it needs. That's all. Ladies and gentlemen, it took only 20 years minus 2 weeks to understand it. And as I told you, I incorporated into this movie structures that were determined under different functional states in different labs, also in ours, of course. So what you see here is the small subunit of bacteria. Actually, the whole movie showed ribosome from bacteria. And you can see that it's made of many components, mainly ribosomal RNA, and many proteins. Proteins, each of them is shown in a different color. The colors have no meaning except that to show you that our proteins are different. What you see here rotating is the small subunit. What remind you, it decodes, so it is, so it is decoding here in the part that connects the upper part to the smaller part here. The molecular weight is almost a megadalton, 0.85 megadalton. The RNA chain in all bacteria is about 1,600 nucleotides, plus minus 20. And it's called 16S RNA because it sediments as 16S. The whole subunit sediments as 30S, that's also the nickname. And there are 20 or 21 different proteins, depending on the bacteria. Reminding you, this is where decoding happens. The large subunit is where peptide bond is being made. And actually not just peptide bond. The next one, the next one, the next one, it means the elongation. And the protection is given by the large subunit. It sediments as 50S. It again is made mainly of ribosomal RNA and many proteins, up to 34. RNA, 3,000 nucleotides, and molecular weight is 1.5 megadaltons. This is the ribosome. So just to remind you again, if this is the small subunit and the lower part, the large subunit, now we look from a different way. They are connected by tRNA that is reading the code, decoding here in the small subunit, and peptide bond formation in the large subunit. Actually, I was mistaken. It's not connected by tRNA. It's connected by itself, by the surfaces, but tRNA fits this way. If we look at the surfaces of the two subunits that associate, the association surfaces, we see that the functional places on them, the functional sites, are RNA, made of only of RNA, protein-free. So this is where the decoding happens. This is the tRNA molecule that combines them. So this is where decoding happens. And this is where peptide bond is being made on the large subunit. And there are 3 sites for tRNA, A, P, and E, as you saw in the movie. All these functions here and here are made only by ribosomal RNA. Just gray, no protein close to it. What does it mean? First of all, I didn't say it earlier, the ribosome is a polymerase. I think you can understand it without me saying that it doesn't make just one peptide bond, it makes the next, the next, the next. So it's a polymerase. And it is a ribozyme because everything is done by an RNA. When I ask young people what they think, young students, what does it mean that RNA is making proteins? So they say the cell doesn't trust that proteins will make themselves, because proteins can make peptide bonds from the point of view of catalytic abilities. So it sounds funny when they say that cell doesn't trust, but I think that it's correct. It is sophisticated regulatory machinery. RNA is making proteins. It is also in agreement with the composition of the ribosome. Most of the ribosomes, almost all of them, are made of 2/3 of them is RNA and just 1/3 is proteins, so it's the main component. Even in mitochondria, where part of the RNA is replaced by proteins, the active sites, the 2 active sites, are exactly the same as in other ribosomes. So what can we learn from that? From the fact that major ribosomal functions are performed by RNA. You can learn that RNA dominated the prebiotic world, and I will discuss this in a minute. And also that RNA, which is known to be a rather inefficient enzyme, usually RNA enzymes are called lazy or lousy, depends how good your English is, but they are known to be inefficient. They can become efficient by nature, mechanisms of nature. Because as I showed you, ribosomes are very, 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 very efficient. So when we look at the RNA composition, and I'm sure you know what is an RNA map, when we look at it, here what I show is the large part of the large ribosomal subunit, we don't see any symmetry. Also, when we look at the sequence of the RNA nucleotides, no symmetry. But once we determine the structure, we found that this region has an internal semi-symmetrical nature, this part, the blue part. And this blue part, for your information, is containing the active site, the peptide bond formation site. So what I mean by semi-symmetric nature is that we can in our mind draw a line here that can represent a twofold symmetry axis, like that every atom here can be found here, as long as we talk only about the backbone, because there is no sequence symmetry. So just the backbone is symmetrical between the two of them, or not exactly, but almost symmetrical. And when we look at it in more detail, this is the upper part of this region, this part, and this is the lower part. The ends of two tRNAs are reaching it. That's where the active site is. So maybe now I'll say in names, the tRNA that is sitting at the site of peptide bond formation is called P, peptidyl transferase RNA, so it's P site RNA, and it sits in what we call the P region. And the A site RNA, that's where the amino-acylated tRNA is reaching the ribosome, is sitting here. The whole region is made of 180 nucleotides in the large ribosomal subunit. So when we look at it, where is it in the whole large subunit? You can see here the skeleton of the whole large subunit. This is the region, the blue and the green. The green is occupied by the P site tRNA, the green tRNA, and the blue by the A tRNA. Here is peptide bond formation point. And the motions that you saw in the movie are made by these two regions, these two hands, which are connected to the region, the symmetrical region, chemically. So you can see that it sits in the center of activities. It also reaches the messenger RNA by the tRNA. So actually it can serve as a signal transmission between all the active sites, because when the A site tRNA has to come here, it has to be empty. It means the P site is already on its way out. And how will the messenger know to move? Only when there is signal transmission. So we think that this is what it does today in the modern ribosome. Or if I can show it in another way, this is where tRNA enters. This is where tRNA goes out. Here is the region, and it is connected through these two hands of the large subunit. If we look at this region, at the fold of this region in all known ribosomes, and I incorporated into here the large subunit bacteria, human, and mitochondria, archaebacteria, eukaryotes, whatever. If we take only the backbone of all this, look how well it agrees. They agree between each other. They are exactly the same. We are looking now at this region that has sort of twofold symmetry here. All of them, all these blue are the A site. And look, I could not, after I made it, here is the chart, I couldn't see which is which. They're all the same. Also in the P site. From point of view of a main chain, they are the same. So we think that this is the prebiotic bonding machine. We call it the protoribosome because it's the same. It doesn't care for evolution. It's the same in bacteria and in elephants. The same everywhere, in flowers, in whatever. So we propose that it's the protoribosome, and we think that it's a prebiotic. It's before life. And what does it mean? that before life there was a machine in the RNA-dominated world, and it's still in each of our ribosomes today, in our bodies or in every contemporary ribosome. So it looks, when we look at it not from top but from the side, it looks like this, the blue and the green, and with the two pieces of tRNA that interact with it. And when I ask organic chemists if they think that this could have been a molecule of its own, they said yes. From point of view of energy, of standing, or whatever, it could be an entity of its own right that does not depend on anything else. So I'm happy with it. It may have been the protoribosome. Just to remind you what is the tRNA, in order that I can talk about the part, what I said here, the part of tRNA that is in this machinery. To remind you, tRNA is a double helical molecule, RNA molecule, that has an anticodon here that is specific for each amino acid that can interact with the codon in the messenger RNA. That's an anticodon loop. And this changes by the amino acid. The structure doesn't change over evolution, almost the same. And the end of it, the other side, the 3' end, is always the same, fully conserved 3' end. C, A, and the amino acid, always. This is all tRNAs. So just to remind you here, this will be A amino acid, A amino acid, and the A and at the P site. So if we look at the ribosome, the contemporary bacterial ribosome, this is the active site in here, and these are the walls of the active site. So we determine the structure of it together with a mimic of the A site. This is the mimic. And you can see the A are inside, where peptide bond has to be made. Everything here is RNA, ribosomal RNA. Please disregard the colors. And one protein is near it, but does not reach the active site. So when we looked at the structures with two tRNAs, here they are. The A site is exactly how we saw the mimic, and the P site is here, and peptide bond will be made here. Like that. So the A site, tRNA, CCA amino acid, P site, CCA amino acid will make the peptide bond here. The gray around this is the ribosomal RNA. So how does the tRNA travels from the A site to the P site? You see, there is here a site, and when the peptide bond is being made, the tRNA will go out to the A site, and A will go into the P site. And you could see it in the movie. How does it happen? It happens by a motion that is made of two parts. Shift of the helical part of the tRNA and rotation of the 3' end. And if you want, maybe we go back to the movie and I will highlight it. Sorry. Highlight what I mean, and then I'll discuss it. So we look again at the same movie as before, now with a little bit more knowledge of ribosomes, if it comes. Okay, so what you see here is the messenger RNA is reaching the small subunit, bacterial small subunit, being connected. Now everything is fine by the factors. The first tRNA is coming to the P site, this here, where decoding happens. Now when everything is fine, the large subunit comes, they can make the ribosome. And the next tRNA and the higher ones will come now from the side, one by one. By the way, this movie exists in YouTube. You can see this, it's there. So they come in, they are being decoded here, and peptide bond is being made here in the large subunit. And we took away the large subunit, so you can see that the tRNA moves sideways, but its lower part is rotating. It comes in this way and comes out that way. Look at the next one, the same. It rotates, this moves, and the other rotates. So so if we have a look again, this is the active site, a mimic, the real tRNAs, and this is the way they are bound. And peptide bond is being made inside this position. So the tRNA motion is sideways, a shift, and rotation. So this sideways goes this way, and the rotation goes around this one. Or if we look at it in the ribosome, the tRNA moves this way, but the lower part is rotating around this axis that I called it semi-symmetric, the imaginary axis, and the whole motion is dictated by the ribosome. These ribs are ribosomal components. We can look at it here. This is the ribosome in gray, and the A to P, the volume that it takes from going to here around this is shown here at the A site and then at the P site. So how does it happen? We start here at the A site, we finish here at the P site. Or if we look at this in more detail, it goes from A to P, and the ribosome is the red that is lit and helps it. So now it reaches the P and it comes again, and we look at the next tRNA. Goes around again, and again, and again. Comes here, makes the bond, goes out, and so on. So if we look at it from bottom, it has here, this is the motion of the tRNAs, CCA, and an amino acid. This is the ribosome, this is the rotation, and there are two nucleotides here that are also fully conserved, and I will relate to them in a minute, from the lower part. And if we look at it from the side, one of these two nucleotides is A at position 2602. Here it is, A at position 2602 goes in between the A and P, and it can have many, many conformations. For instance, with just taking these small molecules, it's called sparsomycin, it can move from here to here, just by having nice contacts to this one. Or if we look at all the positions it can make, if this is the A site and this is the P, it can be like that, like this, like this, like this, like this. We think that it can propel this rotation. It may be the propeller of the rotatory motion. And at the end, of course, peptide bond is being made. From the side, it looks like this. Again, these two nucleotides and the whole motion. We of course, drew it mathematically. And from another way, looking at it, when we look at the volume that is taken by the motion from A to P, we can ask the question, when is the peptide bond being made? At the A site, at the P site, or on the way? And by quantum mechanics, Massa and Carla showed that the transition state, the peptide bond is really being made almost at the P site. This is the transition state. And it has to go all this way. And they showed it in quantum mechanics. To show you the size of it in the whole large subunit, so the large subunit is rotating. This is the size. This is the way it will look with tRNA in a space filling model. And this you have seen before, that's where the peptide bond. It's the same thing, but two different ways to show. And actually, its structure is shown here. You've seen it earlier, and you see it again. This is our suggested protoribosome, as we think, as it existed today and functions today in the contemporary ribosome, and probably similar to this or exactly like this in the prebiotic world. So how was it made? You think that maybe it was made from a dimer. So in the prebiotic world, in the RNA-dominated world, there were pieces like this. They could come together and make pockets. And from the side, we look at this pocket that can accommodate, for instance, amino acid or a nucleotide charged with an amino acid. So we think that this is the way it happened, one of the ideas, not all, one of the hypotheses. And this hypothesis is actually supported by other studies. In order to check it or to prove it, we are trying to create a protoribosome. So we look at this region that I told you earlier, and we produce RNA chains that are either resembling the P site or the A site, and we are trying to dimerize them. So these are actually our constructs, the A site, the P site, since we have to cut the way they are in the modern ribosome. These places here, we can play with. We can do mutations or sort of chemical mutations and so on. So this is what we can play with. And indeed, we try all types of contacts. And we are trying to look what happens when they are of this or that chemical property. For instance, GNRA A minor is a very strong interaction node in tRNA, so we are using these two. And we are also making RNA bits, RNA chains that resemble what we think is the overall structure according to the structure of this prebiotic, what we call prebiotic machinery, that are made like that and supposed to produce three-dimensional structure like this, and we are trying to dimerize them. And to our excitement, we found something we didn't expect, non-uniform tendency to dimerize. We thought all of them are the same, or the nucleotide here, nucleotide there. But no, there is a preference. There is a preference to some that dimerize and some that don't dimerize. It means we discovered pre-Darwinian Darwinism. Only the P site part dimerizes. You know, Darwin talked about animals. We are now talking about parts of molecules, and still they have a preference. And some of them are dimerized, you can see here, and some don't. And I don't want to go into details, but what we found that the P site dimerize, the A site does not. The P and the A do not make dimers, non-homodimers, and also between those that we made according to our wish, the pieces of RNA that we synthesize according to our, or not wish, but the expectations, some dimerize and some not. I want to remind you that only the backbound and the orientation of the nucleotides are symmetrically looking at each other, are symmetrically relating to each other. No sequence symmetry. So maybe in the beginning, the PTC, what we call the active site, was just two P regions. Because in the beginning, before life, there was no reason to have difference between the two sides. It was not that one is in and one is out. Maybe it was all P, and with time, it became specialized for its own needs, and they are now a little bit different. So the backbone of the two regions, A and P, is almost the same, but the side chains are different according to the modern needs. So let's ask a question, a logical question. Why should RNA construct a machine to polymerize amino acids? This action will come out with a competitor that is better than itself. Enzymes, protein enzymes are better than RNA enzymes. Why should it do? So we don't think that really RNA sat there and thought, I want to do it or I don't want to do it. We just think that there was a machinery in the RNA-dominated world, and this afterwards was hijacked by the proteins, and they took it for their own needs. So we are trying to prove this too. And ongoing binding experiments, binding of RNA substrates, yielded very interesting several constructs that have specific binding to RNA, which I won't show now, but it happens. But it also brought us to the thinking, how did the genetic code emerge? The emergence of the genetic code, which is a possible pathway, what I'm going to tell now, not real. Or the question is, what was first, the genetic code or the products? So in our mind, the initial dipeptides could be the substrates of the formation of a next peptide bond, and so on. So oligopeptides could be formed when the amino acids hijacked this machinery. And these oligopeptides that were performing important catalyzation or important tasks in the RNA world, or stabilized the machine that made them, they survived. According to Darwin, the fittest survive. What do I mean? Like an important task is moving around metals. And this is a little oligopeptide, histidine-rich oligopeptide, that can form an internal structure that can be a cage that can move metals, like zinc. Now, histidine, most of the evolution people think that histidine is not an early amino acid. Glycine, alanine should have been earlier. Maybe they're right, but in my opinion, and also in the opinion of the group here in Mexico, histidine is one of the earlier amino acid. Oops. So my good friend that you heard earlier, Antonio Lascano, gave me 2 slides that shows that perhaps histidine is more important than just being there. This is histidine, to remind you. Most of the people thought, until recently, I think that now they change a bit, that the earlier amino acids were just simple like this, not with something so complicated. No, it's not so complicated. It exists in the nucleic acids that make the RNA. So it must have been earlier. And Lascano made a table, which is fantastic, shows all types of tasks in all types of bacteria. And who wins here? Which amino acid wins in this performance? Histidine, until today. Histidine is the amino acid that is either largest or maybe second largest. In all this, you have here the names. In all these reactions. So we think histidine was there. What else could be the reason for needed proteins or small polypeptides? Those that stabilize the machine that made them. And until today, all these positions are not occupied in the in our mind when we cut it out of the ribosome. So here, proteins could have helped. And we based this idea of how this region is in the contemporary ribosome. Here you can see the region in blue and green and all types of interactions within. And they are interacting here at the side with two proteins, L2 and L36, that really holds them together. You can see here L2 and here L36. Just sits here at the end of the symmetrical side. So maybe these were the first proteins or peptides or small peptides that existed and came up. So this suggests that the genetic code was created by or according to its product, which were found fit and useful, and therefore they survived. They led to the creation of a primitive original genetic code which co-evolved together with its products. And I will repeat it. The genetic code co-evolved together with the ribosome and the products, the proteins. Just to remind you of what we are talking, this is the whole ribosome, large and small subunit. This is a piece here that we discuss. You can see it here enlarged, and the tRNAs and the bridge between the two subunits that is associated with it. From top, it looks so beautiful as this, and from the side like that. So this is what we think is the protoribosome. And now we can try to answer the question, what was first, the chicken or the egg? Or in our words, what was first, the genetic code or its product? We think our answer is nothing. The protoribosome was the first. And it made both of them. So I want to tell you some things about the structure of the RNA in the ribosomes, which are really fascinating. And for this, I want to tell you about another fascinating observation. Almost half of the clinically useful antibiotics target protein biosynthesis, mostly by paralyzing ribosomes. And this is because of the fundamental role played by ribosomes. So let's look at this aspect. First of all, the natural antibiotics are the weapons that bacteria from one type is using to interfere with the life of another type, and some of them were increased, improved, extended by companies when antibiotics came into use in the middle of last century. Question is, how do the tiny antibiotics paralyze the giant ribosomes? So ribosomes, bacterial ribosomes, these are the target, are about 2.5 million Daltons. And the antibiotics are 500, 600, less than 1,000. The trick is to bind to active sites. So you see here a skeleton of the large and of the small subunit, and the balloons, each of them represents a family of antibiotics. It means a natural antibiotic after it was improved by companies, so there are some with the same skeleton. These are families. All of them are in active positions, in the decoding, in the bonding, in the tunneling, in the tunnel, last step, but still important, or hinging, these motions that I did by my hand. And for this, we have another movie that we made immediately when we got the structures, together with 2 students of the Art Academy, together with the first one. And look, a small one. How do this small one disturbs protein biosynthesis? It's called edein. Look, look, look. Just a barrier. You don't have to be big to disturb messenger binding. Just this. Set 2. Next one is tetracycline, is more popular, and it disturbs the A site tRNA from binding. Occupies its position. One tRNA is not sufficient, or anyway, tRNA only in the P is not sufficient. These 2 worked on the small subunit. Now let's look at one in the large subunit. Erythromycin, the first, or almost the first one to be used after penicillin, it sits in the tunnel. Only very short protein can be made. 5 to 7 amino acids. That's not enough. So it blocks the tunnel. And the last that you will see is called clindamycin, and it's the cleverest chemically. It goes into the bond. So to remind you, the bond is being made here in the large subunit. Look, in the bond between the CO and the NH. Isn't it clever? So, as I said a minute ago, all ribosomal antibiotics bind to ribosomal functional sites. All ribosomal antibiotics, natural antibiotics, or extended, but based on natural. But ribosomal functional sites are highly conserved. All ribosomes work the same way. And mandatory for clinical use is the distinction between the patient and the pathogen, the bacteria. We don't want to kill the patient. So how does this happen? Even when there is a very highly conserved region, sometimes there are subtle differences, and I want to show you one. So we're looking here at the whole large subunit, RNA in dark green and proteins in light green, view into the tunnel with erythromycin in it. You see it blocks most of it, not all of it, but it blocks very nicely. If we look at it from the side, section through the ribosome this way, tRNA in the PTC sits here. Here peptide bond is being made, it means the protein. If there was no erythromycin, it would have gone through here. For the tunnel, the erythromycin will stop it here. That's the story. How does it happen? So we looked at the structure when we cut the tunnel perpendicular to its long axis here. And we got a pocket. And in this pocket, erythromycin and all his brothers and sisters and cousins and all the family of erythromycin-like bind. And all of them block the pocket. The pocket is made only of RNA. If you can, this is the main formula of erythromycin, or all macrolides. Macrolides is the sisters and the family of erythromycin. All block. Some block excellently, some block very good, some block just block, but all block. And all interact with the tunnel walls, specifically with this nucleotide, A at position 2058, that I made pink. I want to show you how the interaction is. So here, A at position 2056, this is erythromycin, and here the affinity is just excellent, like a very clever chemist sitting in a very important company calculated. But no, it's just made by bacteria. Fantastic, fantastic affinity. This is A in position 58, which is the same in all eubacteria. All eubacteria have A there. And all pathogens are eubacteria. There are many eubacteria that are not pathogens, but all are with A in position 2058. Ladies and gentlemen, have a look at this point, and you will see the difference between bacteria and us. That's it. Bacteria and us, A to G. You know what is RNA, I don't have to explain. That's it, A to G. But when there is a G here, there is a clash. There is a too short van der Waals region to erythromycin, and it doesn't bind. So this is the distinction between patient and pathogens. Another point that I want to discuss is resistance, and I'm sure that you know that resistance to antibiotics is one of the most severe problems in modern medicine. And first, before I go into resistance, people like to blame the doctors and the food makers that they put antibiotics in food or in medicine more than needed. This is correct. But this is not the reason for resistance. It's a reason for growing of the resistance. Resistance itself is a basic process of survival that nature gave to bacteria against bacteria, or bacteria running from others. So in a tribe, there are several studies on it. I will cite 2 years ago, the last one that was published in Science by a group that went to a tribe in Brazil, and they found that this tribe that never got any Western medicine, any Western treatment, in their guts they found bacteria with the genetic apparatus to make resistance. It means it's a basic mechanism that can find in people that who never been exposed to Western medicine. These findings show that resistance to antibiotics is a basic process, and for survival of many microorganisms, regardless of their exposure to modern life, and suggest that microbes have long evolved capability to fight toxins, including antibiotics, and that preventing drug resistance may be more difficult than several scientists think. And it's correct, it's more difficult, it's very difficult. So I just say that bacteria develop specific molecular pathways, and I want to show you one. There are many pathways, I want to show you one that is directly related to RNA. So you remember the selectivity? A in position 2058 and G. This exists and this does not for selectivity. For bacteria, what's the problem? They just change themselves to G. For this is a very simple mechanism. It needs some genetics, but bacteria can change themselves 5 times a day. After a few days, there are hundreds of new generations, and each of them has one mutation. The one that survived is G in this position. So I don't want, I will jump now some, but I want to say clearly that the increasing appearance of multi-drug resistance and the negligible or minimum tries of companies to fight it is really becoming a problem. I think, in my opinion, in short time we will come to pre-antibiotic period where people died from severe infections or even from wounds. So I call it pre-antibiotic. And the World Health Association called it post-antibiotics, but it doesn't matter, no antibiotics. What matters is that recently the world understands it, and even the World Bank estimated that up to almost 4% of the global economy will be needed for this reason by 2050. So it's a problem. And yet, companies do not look for new ones, because they think, as I already showed earlier, that combating resistance to antibiotic may be impossible, unlikely. In my opinion, completely getting rid of resistance will not work, because bacteria want to live in street worlds, and because they are cleverer than us in terms of survival. Not in everything. They don't know mathematics better than us, but they know what they need. That's my opinion. Yet, although companies don't do anything, most pharma companies understand it, we decided that our little lab, we do modest contribution as much as we can. And I won't show you everything that we found and we are doing, because it's much too long, just the RNA part of it. So to remind you, you know that there is a microbiome, lots of bacteria that live in semi-exposed region in our body, about trillions of them, and most of them are eubacteria, and most of them are involved in our well-being. It means when their composition is changed, we can get allergies, Crohn's diseases, diabetics, and so on, autoimmune diseases, so we have to keep them. And the current antibiotics get on them. So if we take antibiotics even a little bit for our health, some of them, if they can penetrate into these regions, they can change the microbiome, and we are trying to get away from it. So I just told this. And I want to say that what I showed about antibiotic binding, like the movie and some papers, this was all done on bacteria that can be good models for pathogens. Pathogens are those bacteria that bring diseases, but not real pathogens. We couldn't do it because of technical reasons, safety reasons. So we learned about the common modes of antibiotics action, but not about, we could not find out, we couldn't explain specificity. Each disease has a disease, each bacteria has another specificity. So for this reason, we crystallized a large subunit of pathogens, staph aureus, multi-drug resistant pathogen, and we looked at the structure. And I won't show you the whole structure, and even not all the lessons we learned, only lesson 4, because I think it's the most exciting one for me, beyond my expectations. So if you look here at the sequence of protein, ribosomal protein L3, in many, many antibiotics, you don't see it. It's not important. What's important is that you can see that only 4 of them have this piece of extension. Otherwise, they are very, very similar in protein L3, this and this. It's a few amino acids. And when we look which bacteria are there are 4 pathogens, all of them. So we looked how it is positioned in the ribosome, and I'll show you that this phenomenon is found also for another protein, L17. So what you see here is the overlay of the RNA of 4 different ribosomes, 2 pathogenic and 2 not, the RNA, and they are exactly the same, and it's shown in cyan. The protein L17, they are in the 4 bacteria, are 2 pathogenic and 2 non-pathogenic. Each of them is a different color. So most of the protein is almost the same in all the 4. But here, there is a piece that is only in one of the pathogenic. So this can be a target for a new type of antibiotics. So I don't think that proteins are good targets. Most of the targets for antibiotics are actually RNA. I just brought it as an example because the real exciting example is RNA. So we look here at helix. It's called H63, in 2 bacteria, pathogenic and a model. Here, they are exactly the same. You can't distinguish. Here, they are exactly the same, here and here. But here, there are 9 additional nucleotides in the pathogen. What is 9 additional nucleotides compared to 4,500? It's less than standard deviation. Nobody paid attention when they did statistics. Even if there are 10 positions like this or 20, there are also positions that there is less RNA, so it escaped the statistics. But when the structure came, look at it. This part is additional for this particular pathogen, staph aureus, this one. So it can be a potential binding site for a new type of antibiotics. And we got very excited about it. First of all, we found several like this. You can see here the large subunit from front and from the side. All these bluish are extensions. I didn't put here the deletions, the shorter. Look at it here. Enjoy. You see on the skeleton of the RNA skeleton of the large subunit, these are the extensions. And all of them are on the surface. Either surface that go into the solvent or surface that sees the small subunit in the assembled ribosome. Here you can see the solvent exposed and those that are involved in the interface. So in a model system, we identified 25 new potential unique sites like this, and we blocked them chemically. In vitro, 16 of them inhibit protein biosynthesis. And you know, bacteria are clever, I said earlier, cleverer than us, but they are not so clever. Because these sites are not involved in primary ribosomal activity, peptide bond formation and decoding, no pathogen contains genes for their modification until now. It means the bacteria still don't understand that they are important. So for the time being, we can use them for new antibiotics. At the end, there will be resistance to them too, but maybe in 20, 30 years, so we have a lot of time to play and to find more and to take cocktails and so on. So for now, these sites can be exploited for the design of advanced degradable antibiotics that are also environmental friendly. I didn't talk about it at all, but I want to say in 2 words, the normal antibiotics, the bacterial antibiotics, the original ones, all of them have a piece, a core that is not degradable. So even if we just take very small part, a very small amount of antibiotics, this part comes out to the sewage. And because it is so small, not all cleaning systems can find them and they can go into the irrigation. And if they are irrigating grass that the cows eat, we get them back by the milk or by the meat or by the cheese. So it's really important. And what we have done, all the so far used for blocking are those that we can improve by improve their performance and minimize their poisoning capacity and make them better killers of the pathogens as we want chemically. So we can use materials that are ecological friendly. Oh yeah, sorry, I have it. So here are the pieces that are not digestible, and this is all what I told you. So far, as I said a minute ago, the inhibition of protein biosynthesis by the newly identified potential sites was achieved by molecules composed of nucleic acids. You all know what is antisense. We just made antisense to these pieces. Simple. Sometimes we added to the antisense oligopeptides to make it more stable and so on, but all of them can be optimized in terms of chemical properties, antibiotics action, and degradability. So the new insight that we obtained from these high-resolution structures are used for genuine pathogens, for antibiotics for genuine pathogens, and they are providing unique tools for suggesting novel sites, I just showed it, suggesting how to improve clinical performance, I didn't show, believe me, there is even a paper, 2 papers, for better distinction between pathogens and useful bacteria, just wonderful. And for the production of pathogen-specific antibiotics. It means like precise medicine, like personalized medicine when the person is a pathogen, a bug. So we are aware of the problematic problems, economical problems. The companies don't like to make new antibiotics anyway because it's too much investment in research and too little output, and there will be resistance and so on. But we hope that the companies will once consider human aspects, not only profit, but this is my personal thinking. What I can talk about more scientifically is the efficient use of pathogen-specific antibiotics means a possibility to find what is the pathogen when a person comes to hospital and has high temperature. Usually it's getting a broad performance antibiotics that will cover everything he may have, but now they will have to find which pathogen it is, and they told me that they are doing it anyway. It also requires it also requires many structures, smooth use of the antibiotics to the specific sites of the pathogen. So we need many structures. Many structures? How? By crystallography? I want to remind you, 20 years, minus 2 weeks, but 20 years for the first ones, and afterwards 2 to 4 years for each. That's much too long. We don't have this time for each pathogen, even if several labs will work on it. So luckily, recently, three-dimensional cryo-electron microscopy became useful, and we are using it. First of all, we wanted to show that we can use it. And we first determined the structure, the 2.8 structure, atomic level structure of the large ribosomal subunit from the parasite Leishmania. Why and how? First of all, what is Leishmania? Leishmania is a parasite, a disease made by a unicell eukaryotic parasite that sits in these sand mosquitoes. And as you can see, 12 million people, 12 million cases were reported last year, and they are mainly in these areas. So Mexico is part of it, but many others, more than half the world. They can be fatal, they can just cause very, very bad diseases, and they're interesting because of it and because of other things. First of all, what are the other things? Why to look at Leishmania? Again come the RNA. This is the human A site in the ribosome between the large and the small subunits, the black and the gray. This sits here, and this is the composition of the RNA in the A site, in human. This is in bacteria. Look what a small difference, just a small difference here. Yet there are antibiotics that are targeting this position in diseases that are caused by the bacteria. And one of them is one of these antibiotics is stopping the genetic problem of jumping over the stop codon. But this is not the point that I want to talk now. One similar one to this one was used in the study that I'm just describing. Here are the differences. And some antibiotics bind only to bacterial, not to human because of it. I can't go into all the details of the structure, but the most important one is this base pair that keeps the G and the C together, but let a lot of flexibility for these two, A and C. So human A site and Leishmania A site now. This is the mosquito disease. The difference is even smaller. Here, C and U. But here there is a base pair and here not. And this is this was the reason why we looked at Leishmania, and I want to show you the results. So within a few weeks, we got this beautiful structure of the large subunit, all the proteins, all the RNA, although for most of them we had also to do the sequence. The sequence was not available. I want to show you the level, the quality of the results. So these are all the all the proteins in the large subunit. You probably are not interested in each detail, but I want to show the quality. We can see side chains. See? In RNA, we can see base pairing. We can see the difference between purines and pyrimidines and magnesium ions. It's possible to do it. It's fantastic. So we want to use this let's forget this. We want to use this method. And I want to show you what we learned about the RNA in Leishmania. So what you see here is the small subunit and the large subunit RNA in bacteria, in eukaryotes, and in Leishmania. Small subunit in bacteria are, of course, larger in eukaryotes. These are normal eukaryotes, and this is Leishmania. That's all. That's the only difference. But the large subunit, in bacteria, there are 2 parts. In eukaryotes, there are 3 parts. And in Leishmania, there are so many parts. And we call it parts because the DNA is continuous. It's just the RNA. So many places that it is cut. And we can see where it is in the structure. Again, the same story in prokaryotes, in eukaryotes, and in Leishmania. Many parts. Where is it? It's like the DNA is one, and these are the positions that RNA was cut. When? When was it cut? So we looked at the structure, and we looked what are the focal points of these cuts between these and these and this. And we found them in these positions, this one, and this one. So we even know that RNA was cut in these positions. We know the enzyme that cut it, but we still don't know why. And we can say that it's actually free enzymes, or free occasions, because the distance between them in this overall structure is over 100 angstroms, so it cannot be the same enzyme the same time. So this is another point that we are investigating. Also, when we look at the RNA, we can see it in very high detail. We can see modifications like methylation or dihydrouridine. Actually, we could see 99 different modifications in the large ribosomal subunit. Most of them were not even reported. So now we are trying to find out why there are these modifications, what they are doing. So this is part of our future. So thank you, and thank the Weizmann Institute that let me work on what was my dream for many years, especially the president that made the decision. He was immunologist, Michael Sella, but when he was replaced by a high energy physicist, he didn't change it, and they were encouraged by these guys. All started with a very fruitful and intense collaboration with Max Planck for molecular genetics in Berlin with their director, Mr. Wittmann, Professor Wittmann. I want to tell you something about Professor Wittmann. He said that he is very tolerant. There were 245 people in his Abteilung, in his department, in 14 working groups, and each of the groups could do whatever they found suitable scientifically, and they could do as long as it was on ribosomes. So tolerating me was not such a big deal for him, one more. He also helped in starting up a group for us, a research unit in the synchrotron in Hamburg. So I had 2 groups, and I want to thank them for their devotion and determination in good and in bad time. Here is the group in Hamburg that was terminated in 2004. And the technical assistants say that only angels can work on ribosomes, you see. The first ribosomes that crystallized were from the Dead Sea. The Dead Sea is a sea with only 2 bacteria and very salty, so the group from Hamburg came to look for bacteria. They had really great time. I want to show you the group in Israel that is still functioning. What you see here is more than the group. The group today is 10 people, but there are some people here from before. I want to tell you that the chief scientist, senior scientist, is Dr. Anat Bashan. She is really running the show when I'm entertaining you. I want to tell you that Ilana found the internal symmetry, and Chen worked on, and Miri worked on making producing what could be the protoribosome. Gil and Tanaya and Giuseppe are doing the antisense and the new positions. Where is she? Zohar and Donna. They determine the structure of the pathogenic ribosome. So everybody has his own, and I'm really very happy with all of them. And I'm sorry. I want to show you Tamar that made a cake for her birthday when I took the picture, and this is her cake. And this shows that in my group, ribosomes are sweet. I want to tell you that my family supported me all the time, my mother, sister, and so on, but I show here only my daughter, who is an MD, and my granddaughter that was a kid at that time, 9 years old. She gave me a prize. She made a speech about me in Paris. She said, my name is Noah, Ada's granddaughter. As you know, she is a very busy scientist, but she always finds time for me. And at the age of 5, she invited me to the kindergarten to explain what are ribosomes. You're laughing? This was so successful that the Weizmann Institute is using this lecture for looking for donations. So anyway, she gave me a prize. I want to show you her prize. That's not her prize, that's a Nobel medal. For me, her prize is as important. The grandma of the year is Ada Yonat. And when I asked her which year, every prize has a year, she said, you have to reprove yourself every year. And if you fail, I take it off the wall. This is the only prize that can be taken from me. All the other prizes are mine, and that's it. But I want to show you, it's still on the wall. Here is a carnival in Israel. He's me and his wife, you see here? She's making a protein. And even small kids became ribosomes. And this is what happened to me by an artist that drew me. If you look at it, he's an artist, not a crystallographer. Small subunit, large subunit, symmetrical axis, and where he thinks new antibiotics have to be. Oops, sorry, thank you.
Questions there, so you can take a seat.
No problem. Thank you.
Thank you.
Was it okay?
It was perfect. There was a lot of new things that I hadn't heard about. Bien, creo que hemos tenido una introducción absolutamente deslumbrante a la estructura, el origen, la evolución del ribosoma, así como a las aplicaciones extraordinarias que tiene en términos prácticos, sobre todo enfrentando problemas de salud a nivel mundial, como es la resistencia a distintos antibióticos. Tenemos tiempo para preguntas. Sí, por favor.
Gracias. I just have one question. Thank you. First of all, thank you very much for your talk. Can you tell us something about the content of molecular, let's say, the molecular content in water, in terms of water, in this peptide forming site?
Inside the ribosome?
Exactly.
The content of water?
Of water molecules.
We never really checked that, but it's quite large. When we bombed the ribosomes with X-rays, they within one shot, they died because there was so much internal motion. So we think it's quite large. It's largest of any other compound, biological compound or molecule or organelle that we know, but we never really checked that.
Okay, thank you.
I'm sorry, we should have. I know water are important, but we can't do everything.
Thank you. May I do another question?
Please.
What's the molecular weight limit of the pre-Darwinism, the pre-Darwinian Darwinism, let's say, as you put?
It's 180 nucleotides, and each nucleotide is 100. So about 2,000, let's the 2,000 Daltons.
Thank you.
Hello. Well, very nice talk. I'm very happy that you were here. So I have a question about, regarding the evolution of the bacteria, because I mean to get out of the antibiotics. So we know that the bacteria are evolving, right? So my question is, what kind of mutations are getting and are erring? You show one that is actually a mutation that is mimicking the human ribosomes, if I'm not mistaken. But I wonder if there are a larger array of different mutations that are not even found in humans.
Now that the many sequences are available, over 1,000 sequences are available. You can look at it there. There are lots of papers and tables. I don't remember by heart all mutation, and we didn't really work on how bacteria evolved. But the data are available. No, not our work, but are available. We paid attention to how from prebiotic it became maybe a cell or something happening within some type of a cell, or at least a position on Earth that reactions could happen. But to call it, we looked at bacteria much too much. We couldn't do everything. Anyway, the level of conservation of this protoribosome piece is the same, 98% same in 930 species. Maybe more. In the beginning, there were only 930 that were available. We used them.
Yes, please.
Buenas tardes. Se dice que, bueno, usted mencionó que las bacterias, ¿en inglés o lo está bien?
Sí, go ahead.
Sí, que las bacterias patógenas tienen una estructura distinta que las bacterias, ¿entendí bien? Sin embargo, en medicina se dice que las bacterias son patógenas en el ser humano cuando estas aumentan su cantidad. Por ejemplo, el Streptococcus aureus es un huésped normal del ser humano. Sin embargo, cuando aumenta su cantidad, se vuelve patógeno. ¿Qué opina usted de esto?
Staphylococcus is pretty non-pathogenic until the population reaches a certain size.
So, first of all, the pathogenicity is not through the ribosome. Ribosome, we don't know about even one ribosome that has to do something with pathogenicity. Bacteria is pathogenic. And we try to get rid of the bacteria through the ribosome, not exactly through where the pathogenicity is, because not always we know it, and for this there are a million other people. We think that the ribosome, we can get rid of bacteria, so we look at the ribosomes. So I cannot really answer you about pathogenicity of bacteria and about, even not about resistance, not a lot. I know that when many antibiotics or a lot of specific antibiotics is used, bacteria hate it and make resistance. And this is just because they want to survive. They're as clever as us, or maybe even cleverer, as I say, they can do this. So to talk about how resistance starts and pathogenicity, I'm not the perfect choice.
Por favor.
Ms. Jonath, thank you very much for your speech. I'm curious about what's the consequence of the mutation in the functionally active sites of the ribosome for the antibiotic resistance. Thanks.
I really didn't understand. You want to know what is the...
Yeah, what's the functional consequence of the mutation of the active sites of the ribosome?
In bacteria.
If there's any modification in the peptide production rate? Or?
Yeah, sure. So the antibiotics that change the active site, there are at least 5 or 6 families like this, and one family, the pluromethylines, have maybe 15 different members. They all cause death of the bacteria by either less proteins in the next generation, or damaged proteins, or wrong proteins. This is exactly what happens. That's fantastic if we can do it, and because there is such a high conservation between the active site of the bacteria and the human, this has to be done very, very carefully. But once it is successful, it's successful.
Por favor.
Buenas tardes. Mi pregunta es, ¿cómo mencionó acerca del RNA mensajero de Leishmania donovani, donde se ve que tiene una secuencia típica en su estructura? Quisiera saber cuándo empezó a cambiar evolutivamente esta secuencia de RNA y por qué, por ejemplo, los kinetoplástidos tienen una, pues digamos, maquinaria molecular atípica, ¿no? Como arreglados en tándem y otras situaciones.
When Leishmania came into life, I don't know. The RNA is not typical. It's just a very long, much longer RNA that is cut. And they are not the only ones. Most of the eukaryotes, I showed the table, sometimes they have less cuts, but all of them have some type of cuts. And we think it happened after, as I really showed on this little picture, after there was already biogenesis, some type of assembly, maybe not fully. Why it happens and how, this is still open for research, but not only in Leishmania, in almost all eukaryotes.
Sí, por favor.
Hello, it was amazing. And I really want to say that it was beautiful watching the molecular differences between Leishmania, that is a early divergent species, and us. And for me, it's absolutely incredible to see that differences because, for example, other protozoa like amoeba and, for example, Giardia lamblia, they have, for example, different structures, not only ribosomes, but, for example, proteins that don't have the same, how could I say this thing?
Sequence?
The same...
Sequence?
Yes, a sequence, but have the same structure. And for me, it was totally amazing.
Thank you. I do think that ribosomes are amazing.
Paco, Dr. Bolívar, perdón.
Thank you. I really enjoyed your talk. I would like to see if you could explain a little further how are you expecting to use the differences that are present in the pathogenic bacteria at the level of the different sequences to direct specific targets for the pathogens.
So I showed how we want to construct them, how to direct them and get them into the cell. This is what we are trying to do now. Okay. That's not simple, but there are examples that antisense could get into cells, and chains of nucleotides, so of nucleic acids, of amino acids. We are looking. There are some that are doing it with phages. There are some that are binding a specific chemical compound, but we know that antibiotics penetrate into cells. So we are trying to learn from the bacteria how to do it.
Thank you.
Sí, la señorita que está aquí.
Hello, I have a question. Have you ever studied the protein components of the ribosome as a possible target for drug design?
So I showed one.
Yeah, but I mean, like, if you have like a study further that.
Just a second. First of all, this whole species specificity could be started only after we solved the structure of a pathogen. And this happened 2.5 years ago, not even 2.5, 2 years ago. So it's not a long story, it's new. But second, whenever I talk to people that are deeply involved in proteins, they say, this is just a loop, it will be flexible, it will go back, won't have a structure. They always try to tell me it's not a good idea. I don't always listen to them, but I'm sure that RNA is better. And all the normal antibiotics that are targeting ribosomes, I don't talk about other proteins, go to the RNA. Must be something in it. We have to study it from this clever bacteria.
Well, I have one question more, and maybe this question is outside your expertise, but I would like to know your opinion about. Well, do you think the existence of some molecular machine like the ribosome, no, that is capable of synthesizing with very accuracy and efficiency polymer like a protein, do you think their existence prove that we can produce another molecular machines to synthesize other molecules or polymers that are even non-natural? I'm telling you this because in the nanotechnology dream, at least the earliest starts of the nanotechnology by Eric Drexler, he was saying that the nanotechnology, the manipulation atom by atom, molecule by molecule, is proved because there is something that exists, that is the ribosome. But he was dreaming to produce some ribosome-like machine, but instead of proteins, produce like something like, for example, diamond. So, but I mean, he tried to extrapolate, like Da Vinci, that he was saying that we can fly because birds can fly. So I don't know if we can produce some ribosome-like machine to produce something really interesting and novel.
Okay, so your question has many questions, sub-questions. First of all, why to have non-normal proteins or non-normal output of a machinery? So many people say they can make better chemical entities that can do better folding, better cutting, better targeting. It means not normal proteins. So the level to, if you want to use ribosomes, the level to start it is to cheat the tRNAs, the synthetases. Whatever you put on tRNA, if it's not too large and you have an anticodon loop, the ribosome will take. In this case, it's very stupid. It doesn't check unless it's too large or L-amino acid. Then there is a police nucleotide that doesn't let, but everything else you can put in. And this has been used, especially in La Jolla, but in many other places, a lot to make non-normal ribosomes that are more stable or more distinction or whatever property they wanted. So that's one way. Second way, there is even a company in Germany, in Berlin, that makes whatever you want, proteins or non-proteins, and they call it cell-free. You know what they have in the cell-free? Ribosomes. So they take the ribosomes out of the cell, and then they have machinery, and they do what you want as long as the bond is being made. And they cheated their cell-free system. They can take even non-tRNAs or modified tRNAs. So that's one side of your question. The other side, if we want to make a better ribosome that can work under different conditions, maybe. If you tell me what is the condition, maybe I can suggest. But so far, there is no man-made, scientist-made machinery that can beat the ribosomes in terms of efficiency, productivity, fastness, and most important, correctness. So if you want to take a chance, if you want to make I don't know, an elephant that has his legs up, instead of stands on the ceiling. Maybe you need a specific protein for this. So proteins are much easier to be changed and to give them new tasks. Ribosomes will make the peptide bond. In this, I say that ribosomes are cleverer than us and so on, in getting a chain of instructions like messenger RNA. They are very stupid. They'll do whatever is there.
Sí, por favor.
Buenas tardes. Mire, a riesgo de hacer una pregunta tonta, desde el jueves que he estado asistiendo a este evento, hasta ahorita no he escuchado ninguna relación con estas moléculas entre vivas y muertas, conocidas como virus. Entonces, yo, a riesgo de hacer una pregunta tonta, pues quisiera saber qué está pasando con los virus en este evento, desde el jueves que está asistiendo, en las diferentes exposiciones. Pues parece como que los virus no existen, o qué está pasando, no tienen importancia, no hay alguna razón para hablar de ellos, o no es su evento. Gracias.
He likes to talk.
Yeah, basically.
So viruses have no ribosomes. So when they invent a cell, either bacterial cell or human cell, elephant cell, whatever cell, they are clever enough to take advantage of the ribosomes there and tell them, to give them the genetic code that they have. They have some promoters, and then the ribosomes of the cell make new viruses, and in this way also sometimes kill themselves. So I didn't talk about all the thieves and all the criminals.
Yo, si me permiten agregar a propósito de los virus, de hecho estaba contemplada la participación desde un principio de la doctora Susana Manrubia, que es una especialista en la evolución de virus de RNA, pero que además de eso tiene 2 gemelitos, y era un poco complicado para ella asistir. Los virus han estado subyacentes en toda la discusión, y si no los podemos incorporar en un árbol evolutivo tradicional como los que se hacen con organización celular, por ejemplo el esquema de los 5 reinos de Lynn Margulis y Caroline Schwartz, es porque los virus no tienen una organización celular. Puede irse hacia las filogenias, hacia los árboles evolutivos basados en secuencia, y entonces un problema con el que se habían topado desde hacía muchos años los investigadores es que en particular los virus de RNA están evolucionando tan rápidamente que no tenemos manera de encontrar secuencias conservadas que permitan comparar grupos muy distintos. Por ejemplo, el virus del VIH con el virus del Zika, con el virus de la influenza, con el virus de la viruela, que es un virus que tiene el material como material genético DNA. Sin embargo, en los últimos años ha estado aumentando el número de estructuras terciarias de distintos componentes de los virus que están disponibles en las bases de datos, y el año pasado 3 grupos distintos demostramos que se podían construir filogenias de los virus basados en un marcador en especial, en la estructura de las polimerasas DNA polimerasas o RNA polimerasas monoméricas, y claramente en todos los casos ese componente de los virus desciende de componentes celulares, lo cual implicaría que vienen después que aparecieron las células. Permítame nada más agregar que el próximo 9 de junio, como parte de los viernes de la evolución que se organizan en el Colegio Nacional, hemos invitado al doctor Francisco Varela, Santiago Varela, de el INER, del Instituto Nacional de Enfermedades Respiratorias. Santiago Ávila. Sí, Francisco Varela era un físico teórico que ya se murió. No lo podemos invitar. Bueno, lo podemos invitar, pero no nos va a hacer caso. El doctor Santiago Ávila, que va a hablar sobre la evolución de el virus del SIDA, de la inmunodeficiencia humana, de las variantes que tiene y de la interacción con la estructura genética que tenemos en México. Entonces, los virus ahí están. No sé, sí, por favor.
Good night, doctors. Dr. Jonat, a fabulous lecture. You show a little of your work in the study of the structure of the ribosomes, relationship to the design of new antibiotics. However, exist other mechanisms of resistance of antibiotics like efflux bombs and hydrolases. Have you studied the interaction of the structure of the ribosome and these kinds of molecules?
Did you understand? I'm sorry, I didn't understand the question.
Sorry, the last part. Do you study the structure of the ribosome and find some structures that only are present in some pathogens? Okay, I'm right?
In some pathogens.
Sí,
en algunos patógenos.
Sí.
We ourselves determine the structure of ribosomes in 2 bacteria. Now it's in 3 bacteria and in Leishmania. But there are now, when I started, everybody did not believe it's possible. Now there are about 30 structures of all types of ribosomes in eukaryotes, in mitochondria, in prokaryotes, in archaea.
Yes. Sorry, encontré mi error en la pregunta, perdón. Han estado estudiando la estructura de los ribosomas para diseñar nuevos antibióticos. Ahora, mi pregunta es, si han estudiado en esos nuevos antibióticos la interacción de la estructura del antibiótico con otras proteínas como son las bombas de flujo o las hidrolasas que pudieran interferir en el funcionamiento de los antibióticos en el patógeno.
So I can answer the first part of the lecture. We didn't study the ribosomes because we wanted to understand antibiotics. We studied the ribosomes because we wanted to understand protein biosynthesis according to the genetic code. When I started, it was known that some antibiotics are stopping the function of ribosomes, but even in my best dreams, I could not imagine that I will understand how they work. I could hardly dream about part of the ribosome. So the antibiotics is a bonus for me. It's not the reason I went into these studies. And it continues to be the practical part of what we are doing. I much more want to understand the protoribosome than how many drugs are working, because on this there are millions of people that are interested and the companies. And what I can contribute is just from what I know best, which is the ribosome. So I'm not looking at how antibiotics are interacting with others. For this, there are doctors and companies. I'm just trying to understand better how ribosomes work.
Thank you.
Bueno, permítanme aprovechar del privilegio de ser moderador de esta sesión para hacer la última pregunta. Allow me to make the last question of this session. So let us go back to your first lab, which is how the ribosome works, and this fascinating discovery about internal symmetry in the protoribosome, or which may be the protoribosome. If one assumes, and I think we have very good biological reasons to do so, if one assumes there was an RNA world, then clearly, the appearance of histidine can be understood as an outcome of an RNA world.
Yeah, sure, because the RNA well needs the imidazole.
Right, so in that case, it will be perfectly evident that one should not look for prebiotic synthesis of histidine or histidine in meteorites, but let us just look at the biological description of the processes then allow the evolution of such a complex and peculiar amino acid from an RNA world.
So the different opinions about be it there was an RNA world, or RNA-dominated world, or this type of world, or what comes from outside, and so on. This is a reason for a lot of research, some of which was just mentioned. I'm lucky. I came in when there is already pieces of RNA. It means all the other questions are questions for me too, but it's not something that I'm studying. So from this point of view, I already have pieces of RNA. I'm trying to make a proto-ribosome, and then the whole ribosome is made by nature.
Bueno,
so
creo que
it
con
was difficult enough, don't worry. Lucky, it was difficult.
Creo que con esta elegía a la estructura, las propiedades, las características del RNA, podemos dar por concluido estas jornadas organizadas por el doctor Bolívar, por el doctor Juaristi, por mí mismo, como parte de las actividades que hacemos en el Colegio Nacional. Yo quisiera decir que es absolutamente maravilloso ver el interés de muchos estudiantes de licenciatura y de posgrado que han asistido a estas charlas. Creo que es un público totalmente inédito para nosotros en muchos sentidos y a nombre de todos ustedes, a nombre del Colegio Nacional, a nombre del doctor Bolívar, Juaristi mío propio, le queremos dar las gracias a la doctora Jonat por su espléndida presentación que demuestra que las fronteras entre ciencia básica y ciencia aplicada son absurdas y que lo que hay es buena ciencia y punto. Y desde luego darles las gracias a ustedes por su presencia. El próximo viernes vamos a tener una sesión totalmente diferente a estas del mundo del RNA, en donde vamos a discutir en el contexto específico de las declaraciones xenófobas, racistas de la presidencia actual de los Estados Unidos, la movilidad de los científicos como un componente esencial del desarrollo de la investigación y de nuestro conocimiento de la naturaleza. Muchas gracias. Thank you so much, Ada.